Sponsored by RxCardClub.com. Get a free prescription drug card and save up to 85% on prescription drugs. RxCardClub.com, a new approach to study treatment resistance in high-grade serous ovarian cancer, clone-specific mutations and structural variations in scWGS. A scWGS-based copy number heatmap for patient OV004. Each row is the copy number of a cell. Cells are ordered according to a MEDIC2 computed single-cell phylogeny, shown on the left. B. Clone pseudobulk copy number at 10 kilobit resolution on CHR. 17 for clones A and B. Truncal variants, TP53 missense and deletion, are annotated in purple. Clone-specific duplications and SNVs are annotated in red and yellow, respectively. C. Phylogenetic trees annotated with cells that have support for variants shown in B. D. Clone pseudobulk copy number at 10 kilobit resolution on CHR. 8 for clones A and B showing divergent chromothripsis on CHR. 8. In B and D, notable regions that are different between clones A and B are highlighted in gray, and the absolute difference between copy number states is shown in the top track. Credit, Nature, 2025. DOI, 10.1038/S41586025095-09580. Break several factors make ovarian cancer particularly challenging to treat. This is largely because the cancer often spreads at a microscopic level within the abdomen early on, resulting in diagnosis at an advanced stage. Additionally, while initial treatments with surgery, chemotherapy, and maintenance therapies are successful for many people, most advanced stage ovarian cancers eventually come back. Break Now. A research team at Memorial Sloan Kettering Cancer Center, MSK, is aiming to find new ways to stop the most common and deadly form of ovarian cancer, high-grade serous ovarian cancer, from recurring with the help of a method they developed for tracking the evolution of treatment-resistant cells in ovarian cancer using blood tests. Their findings, which are published in Nature, may aid in development of new approaches for identifying and ultimately targeting the specific subpopulations of cells that cause recurrence. High-grade serous ovarian cancers (SOCs) contain a diverse array of cells, some of which will be sensitive to our best treatments and some of which will be resistant, says study first author Marc Williams, Ph.D., a postdoctoral researcher who uses computational techniques to study cancer evolution. Existing methods to monitor these cancers, however, don't distinguish between one population and the other. So, we decided to build one that could. Tracking changes in ovarian cancer over time using CloneSeq-SV break to look at the differences between cells within an ovarian cancer, and how their composition changes over time. The research team developed an approach they call CloneSeq-SV. The method combines single-cell whole genome sequencing and targeted sequencing of structural variants, which are large-scale alterations or rearrangements in DNA. This allowed the scientists to monitor the cancer's evolution using initial samples taken during surgery followed by an ongoing series of blood tests and to track which tumor subpopulations survive treatment and which die off. Essentially, we were able to use these structural variants as molecular barcodes, which allowed us to track tumor cells subpopulations in the bloodstream, says senior study author Sohrab Shah, Ph.D., who heads MSK's Computational Oncology service and serves as director of the Halvorsen Center for Computational Oncology. The study analyzed blood samples taken from 18 patients with SOC from the time of diagnosis through the recurrence of their cancer. Using this new method, we could see that the cells that are resistant were present at the time of diagnosis, and that they were able to multiply as cells that were more sensitive to treatment died off, Dr. Shah adds. The study would not have been possible without extensive collaboration and teamwork between computational researchers and the surgeons, pathologists, and oncologists at MSK who specialize in gynecologic cancers, led by Chief of Gynecologic Medical Oncology Carol Agajanian, MD, Chief of Gynecology Surgery Nadeem Abu-Rustum, MD, Director of Gynecologic Pathology Lora Ellenson, MD, and Director of the Gynecology Disease Management Team Research Laboratory Britta Weigelt, Ph.D. How CloneSeq-SV could help future patients. CloneSeq-SV allowed researchers to identify a clear pattern. The subpopulations of cancer cells that rise to prominence during recurrence have distinctive features, including amplifications of certain cancer-driving genes, oncogenes, chromothripsis, where the chromosome shatters into many pieces and is stitched back together haphazardly, and whole genome doubling. Break together. These findings provide new opportunities to develop treatment strategies to attack vulnerabilities associated with those features, Dr. Williams says. He points to an example from the study. One patient had an exceptional response to trastuzumab deruxtecan, a drug that targets the oncogene ERBB2, and remained disease-free years later. The analysis revealed why that was the case at the outset. The tumor contained a mix of cells with and without an amplification of ERBB2, but then first-line treatments eliminated the population of cells without the amplification, ultimately resulting in a cancer with many extra copies of ERBB2 during recurrence. And because we have a drug that specifically targets ERBB2, this evolutionary shift left the entire tumor sensitive to a new, targeted treatment, Dr. Shah says. Next steps for the research. Next steps for the research include studying more patients to identify more patterns that could guide treatment strategies, as well as capturing a richer picture of tumor cell diversity by collecting more tumor samples during follow-up procedures. Moreover, the same approach might be applied to other types of cancer that are also characterized by high levels of variability, genomic instability, the researchers note. More information. Marc J. Williams et al., Tracking Clonal Evolution During Treatment in Ovarian Cancer Using Cell-Free DNA, Nature, 2025. DOI, 10.1038/S4158602509580. Provided by Memorial Sloan Kettering Cancer Center. Citation, A New Approach to Study Treatment Resistance in High-Grade Serous Ovarian Cancer, 2025, October 1, retrieved 1 October 2025 from https://medicalexpress.com/news/2025 10 Approach Treatment Resistance High-Grade.html. This document is subject to copyright. Apart from any fair dealing for the purpose of private study or research, no part may be reproduced without the written permission. The content is provided for information purposes only. rxcardclub.com. Get a free prescription drug card and save up to 80% on prescription drugs.